Hello, everyone, and welcome to another Dolphin Communication Project Deep Dive. Tonight, we are joined by Dr. Amy Hirons. She is a professor and oceanographer at Nova Southeastern University, and she's going to chat with us tonight about the ways in which the largest ocean basin in the planet is actually a collector of history. If you are listening live, you can go ahead and use the chat function to submit your questions. You can submit those questions to DCP or to Kathleen, who's also monitoring the chat with me. And if you're listening recorded, we hope that you enjoy tonight's talk. Dr. Hirons is at Nova Southeastern's Oceanographic Center, and of course is a friend of DCP. Her lab is the Charismatic Megafauna and Oceanography Lab, which is a part of a larger research consortium, I believe, the study of environmental conservation through leading edge research. And we are super happy to have you here tonight, Amy, and we're looking forward to the talk. So with that, I'm going to pass it over to you.
Thanks, Kel, and thanks, Kathleen. I appreciate the invitation, and I'm very happy to be here this evening. So let me just make this a little bit broader. There we go. How's that?
Excellent.
Okay, so my name is Amy Hirons. That is true. She wasn't lying. I am an oceanographer. I was born here in the mainland, but grew up in the Hawaiian Islands and didn't move to mainland United States until I started graduate school doing my master's. And it was there I went and sort of explored the Rocky Mountains because I sort of had this, I guess, a romantic image of vast amounts of elevation and land, and when you can traverse around your island, your home habitat, in less than 3 hours and really not drive in a straight line, you appreciate those kinds of things. And the reason I'm telling you that is because it was one of the things that probably drew me to going to Alaska, where I did my doctoral work. And I can give you the fancy title of it. It was basically using stable isotopes ratios to study the trophic dynamics of Alaskan water pinnipeds. So let me translate that as basically saying that I use stable isotope ratios. It's just a sort of a biochemical way of assessing, in this case, what are seals and sea lions actually eating in the environment? And the reason for that is because at the time in the 90s, these animals, many different species, were undergoing these really dramatic population declines. And many things were looked at. Was it bacterial infection and disease, interaction with fishing communities, commercial fisheries? And then it sort of was starting to hone in on the question, is it food? Was it food related? So in the span of my time in Alaska, I basically learned to use a lot of different techniques to work with organisms that by their nature aren't really conducive to working with. And for the most part, that's marine mammals. So I have learned to use different parts and pieces and information of wild animals and even plants to tell a story as to what's going on in their environment. The majority, but not exclusively, of my research tends to focus on the Pacific Basin. I tell people I'm a Pacific girl, and that's where I orient. My work goes pole to pole. It transfers from the Arctic to the Antarctic and points in between. Some of the major regional locations right now are in Russian and Alaskan and Canadian waters, so the North Pacific to the Arctic Ocean, the mid-Pacific Ocean basin to the Hawaiian Islands, Micronesian Islands, We have a large study site going on in Peru. The site itself is small, but the magnitude and the number of studies is pretty large. And then we currently are collaborating with others on work in Antarctica. So that's where the Pacific Ocean Stories part comes into play. Another way that we could actually, I'm trying to advance here and it's not advancing. Bear with me a second. Another way that we can retitle this is, how do you use biological organisms to release really expensive equipment? And doing that is predominantly in my lab through the study of energy. And energy could be referred to or thought of as waves and currents. But for us, energy is actually, what does it take to survive? And that means to swim, to hunt, to reproduce. And all of that requires food, just like with any human and any other terrestrial organism as well. So this image here is just to give you an idea of just a small bit of the complexity of what goes on in a marine food web. It's a lot easier to stop and think about, what did you eat today? And let's take a moment and just say that maybe you ate a fish sandwich. So there was some kind of bread or bun. There was fish. There was perhaps some vegetables on it. There might have been a sauce, which might have had eggs or oil in it. So it could have been easily a mixture of different plants and animals and different components put together to make that sandwich. Well, now if we think about it in terms of, let's imagine you're a marine mammal, you're a dolphin, for example, and you're not actually tossing together a Caesar salad in the ocean, but what you're doing is saying, great, here's a school of fish, and I enjoy that fish, and that fish gives me a lot of energy, makes me feel good. So now you, as a human, think about something that you eat that makes you feel good, that maybe not only tastes good, but that gives you energy and you feel energetic and not sluggish and feeling like you need to take a nap. And it is a multitude of organisms, just like we're showing here on this kind of depicted food web. So a lot of the organisms, it turns out that we work on and use their tissues to work with are what we call apex predators. So predators meaning they hunt, and they tend to be at the top of this food web. So they tend to be larger organisms. And in many, but not all cases, they can certainly be marine mammals, much like I'm showing here. Whether you are filter feeding microscopic plants and animals, like the bowhead whale here in the upper left, or a manatee up on the upper right, which is an herbivore. They're just grazing on plant material for the most part. But if we look at the lower two images, we have a sea otter on the left and a stellar sea lion on the right. And one is eating bivalves, or the mussels of some kind of shellfish. And that sea lion is definitely eating a very large walleye pollock at this point in time. We think, wow, that's pretty massive. But then we can even go maybe a step further, and we call it a trophic step, or part of instead of a food web, maybe in school you learn more of a food chain. And then we can think about a marine mammal that may actually be foraging on other marine mammals, like our Orcinus orca, also known as killer whale. So as I've mentioned, our work goes on pole to pole. This is one of our images from an ice flow off the Northern Peninsula in the Ross Sea in Antarctica. So what I want to do is, when I look at this as an oceanographer and I'm on an ice floe, or I'm on a ship, And I'm thinking about I'm a tiny little speck in the grand scheme of things. How can I get more information about this environment? How can I learn more? And I can't do it alone. And you, oh, well, you have other scientists, or you have ships or fancy equipment that is very expensive, much like my secondary title. And that in itself also doesn't tell us a lot of what's going on, because there can be great influences. And one of those in particular is human, or what we refer to as anthropogenic, meaning human based or human sourced. So it could be things much like I'm showing here, is not only do you have food or a source of nutrients and energy, but there can be a variety of contaminants that can enter the environment as well. And they can be naturally sourced. They can be from volcanoes in the interior of the planet, but they can also be from byproducts of the industrial development and mining and agriculture. And that serves as a secondary benefit, if you will, of studying food webs and studying whom's eating whom, is that you're also ingesting a variety of different kinds of contaminants and in different quantities. So we're, by studying one thing, which is sort of a food web interaction, we can actually get information not just about food or what's depicted here in the lower left corner, the trophic transfer, but we can also look at, to the lower right here, a suite of different chemicals. PAHs, for example, are polycyclic aromatics. They're basically petroleum products. And whether or not you know this, the vast majority of plastics, no matter what kind of plastic in the world it is, are produced using petroleum products. We can get PCBs, which are mostly insecticides, pesticides, to BBDEs, which are flame retardants. Okay. And all of these things can be affected by the natural environment, which is the upper part of this image. What's actually going on? We've got UV radiation, we've got wind effects, we've got wave actions. And all of these things can, if you have particles, say a solid piece of plastic in the ocean, can get broken down into smaller and smaller pieces. And then it can start to degrade to its chemical components. So within seemingly studying just one area of interest, being food webs or what you're eating, we probably all know the expression, you are what you eat. You really are, except it's more you are what you actually incorporate in your body, for better or worse. And it's not just what's anthropogenic or what humans are inducing directly. This image here is a photo I took on the north slope of Alaska. So that water body you see out there is actually the Arctic Ocean, where it meets the Bering Sea in the north part of the Pacific Ocean. And if you look here, you can see what looks like grasses along the top, and then this dark area, which seems to be, dirt, soil. But then you see this sort of white area here, and then you see it here in this. And that's actually frozen water. This is what we call it's basically it's ice. It's what we refer to as permafrost, meaning permanent frost. And depending where you are, how high a latitude you are, meaning how close to the poles on the planet you are, the more permafrost you're going to get and the deeper it's going to go. Well, what's been happening, as I'm sure we've all heard over the last several decades in particular, is that the planet as a whole is warming. And so we think about sea ice not developing and glaciers melting and erratic weather events. But what that's also leading to is things like thawing of permafrost. And you go, okay, well, it's water. It's going back into a body of water. But what's also happening here is that over the decades, whatever's been incorporated in this living material, in this case, the plants, is also being dumped back into the ocean. And that can be a variety of chemicals that humans have put into the environment that have been sort of locked away in this frozen piece of ground. But now that it's starting to thaw and break apart, it's falling into the oceans. And then the currents and tides will transport it and mix these water bodies. By the way, we're losing meters. So the equivalent of, at various times, 1/2 to 3/4 of the length of a football field. We lose coastline every year. And that can have direct ramifications to the organisms in the environment. In this case, this is a sea lion that has died and washed up on the shore. Now, why it is a loss, yes, all life lost is something that is tangible and that we're no longer going to have, but we want some benefit to come from that. So in this case, this animal is recently deceased. And if we can find animals like that, we will go in there, or our collaborators, because I'm obviously not, well, you don't know that, but I'm sitting here in Fort Lauderdale right now. So I'm obviously not in Alaska, where if this animal washed up there or along the Oregon coast, I'm not there to take advantage of this opportunity to sample some of its tissues, but I have collaborators around the world that do. This image here is going to depict information that you might be more familiar with. So if you look at the axis along the right-hand side, you can see from top to bottom, there's humpback whale, fin whale, sperm whale, shearwaters, sea lions, a variety of marine mammals, large fishes like yellowfin and albacore tuna. There are sharks and sea turtles and marine birds and squids. All this information is plotted together. This is what we call tracking information. And it's been gathered through instrumentation that's been deployed. And in this case, over 15 years worth of information is gathered here for the Pacific Basin predominantly. And if you look along the very bottom, it'll show you some of the colors and individual species and their distribution, like tuna here on the far left in the blue. And then we have seals in sort of golden color, and Galapagos sharks in the red, and sooty shearwaters in the purple, and penguins here in the green. And all of this data, this tracking data, has been laid on top of each other to create this one composite image. What does that tell us? It looks very complex, but it also says we're not static. These organisms aren't static. They're moving around, whether they're moving and traveling in a migratory fashion, meaning they're going to one location and returning to a set location, or they just have great travel movements. They're wanderers of the ocean. And they're not just wandering, but they're searching for food, and they're eating along the way. Just like you would take a road trip, you would stop and maybe get a bite to eat. And then you'd travel down the road, and later on you'd stop and get a bite to eat. You're incorporating food, and you're incorporating information based on that food in those particular areas. And so one of the things that I do, because I don't want to have to rely on finding dead animals. And by the way, if you're talking about a marine mammal, and actually almost every organism we're talking about here, they're highly protected. You can't just go out and kill an animal. You can't go out and just approach these animals. So you've got to get a lot of permits. You've got to get a lot of cooperation. I'm working globally. I can't be everywhere at the same time, and I don't have those kinds of resources. I'd love to be able to say, yeah, I'm going to catch a plane for Patagonia tomorrow, but I just don't have that. So what I thought about doing ever since I moved to Alaska is, how can I use a portion of the animal or even plant that does not kill it, that does not harm it? And the reason I'm showing this image of this fur seal is because one of the things that they can part with are whiskers. And you can see that there are multiple whiskers on the face of this fur seal. Whiskers are just like our fingernails, our hair, they're made of keratin, and they're continuously growing. That's the other wonderful benefit of this, is if I can pull a whisker, and I pulled a lot of whiskers, I get a timeline, a series of information. It's just like if you were to pull a hair from your head, assuming you had some longish hair, it took a while for that hair to grow, just like it took a while for that whisker to grow. And the entire time it's growing, it's locking in information about what it ate, for better or worse. So it could be a contaminant or it could be nutrient, but it also is providing information about not just what it ate, but where it ate what it ate. So this is an example when I said I would use a biochemical technique. This image here represents a whisker. So just to quickly orient you, I'm not going to spend a lot of time here. If you look along the x-axis, the horizontal black line at the bottom, it runs from 0 to 40 centimeters are the units. At 0, that's the base of the whisker in the face of the animal. And the older part of the whisker is out here at 10, 15, 20, 22 centimeters. Really quickly, what this is saying, you can see it sort of oscillates. It's got that wavy pattern back and forth in both the blue, but certainly in sort of the mauve color, the reddish color. What that ultimately was telling me after I figured it out is that this animal is moving back and forth seasonally from one body or portion of the body of water, in this case, near the Bering Sea, where I've written June here at the base of the whisker. And this is where it was spending its winter. And then here, as it gets high, is summer, and then back down to winter, back down to summer. And in this case, this animal's whisker is representing nearly 7 years worth of information. Now, whiskers are great, and they are just one tissue that we can use to give us an idea of what's happening. Another one are teeth. Now, you think, how in the world am I going to get a tooth out of particularly a wild animal, which I bet you can guess the animal on the upper left here is not a wild animal. It's what we call an animal in professional care. It can't live in the wild alone. But there are animals, whether they've died, but whether they can also sacrifice a tooth. The tooth that you see on the upper part of the image in my hand is a harbor seal tooth, which is a much smaller animal. It's usually less than about 6-7 feet maximum in length, versus this canine tooth from a stellar sea lion on the bottom part of the image. This part of the tooth that you see here that I'm outlining in red, this is what's exposed. It's just like our teeth, what we see when we smile or touch our teeth. That is just the enamel. That is just the upper part of the tooth. All the rest of this is root, and it locks its way into the jaw. So now look at that. That stellar sea lion canine tooth, that pointed tooth like we have, is strongly embedded in the jaw of that animal. Why does it need such a massive root? Because they're using their mouths and the teeth in their mouths like we use our hands. They don't have the luxury of having an appendage that they can grab their food and hold on to it while they take a bite and chew. They've got to be able to grab onto their prey, their food item, and hold on to it while it's struggling to free itself. But what we can do is when we get access to teeth, is we cut the tooth longitudinally. So from the tip of the tooth all the way to the base of the tooth. And this is a thin section. This is what we call a photomicrograph. And if you look at the image on the left, this upper part that's labeled enamel, this is the part of the tooth that's exposed, like I just showed you in the previous image. The rest of this is considered root. What we've done is split that tooth in half longitudinally. And if you look here and close on the right, you can start to see there are these dark and light colored bands all along the length of this tooth. And those are what we call growth layer groups. They're basically like tree rings. And every year, a layer is laid down. So the more this tooth fills in, it tells us how old the animal. In this case, this animal is 13 years old. But we can also biochemically analyze each of those growth layer groups to get an idea of where the animal might have been and what it was feeding on in a particular year. Another type of tooth or modified tooth is a tusk. And these are narwhals. These are also a marine mammal. And they have an upper canine, like the pointed tooth I had shown you in previous images. And it sort of modifies and grows outwards. Okay. And this is what a narwhal tusk looks like. And you can see it sort of grows spirally. Well, we can do the same thing with a narwhal tusk that we would do with what we'd call a more normal tooth. And we can split it in half and look at that chemical signature. This is an aerial image of a bowhead whale in the Bering Sea. So these animals use baleen. They don't have teeth in their mouths. They have a series of plates that are side by side by side, and they use them, and again, same material like our fingernails and our hair. It's keratin, but they can actually filter out microscopic organisms. So hopefully no one's upset by this image. It's hard to get an image of a whale on land that isn't one that has been killed. And this has been killed for food. Now, there are very few groups in the world that can do that, but these are indigenous groups, meaning these are people native to a certain location. In this case, this is the North Slope of Alaska. And so these are Native Americans. And they are allowed and permitted, if you will, by the federal government to have so many takes or kills a year, but they use and eat the entire animal. But what you can see here on the right-hand side of the image is this whole series of baleen plates that they use. And you can see it sort of sweeps down and it has some fringe. That collects all the food, the little microscopic organisms that these animals eat. So baleen is one tissue we can use. We can also use a variety of other body tissues. So I said that the indigenous peoples, the Eskimo in this case, would actually utilize all parts, the whole animal, bones, fat, etc. They don't have regular grocery stores like we do and could go buy a carton of milk or get a pizza. They have to hunt and gather all their food. So I thought this would be interesting to see. This is a cross section. If you look from left to right, see this thick, dark layer? That's the skin of the whale. very dense and thick. And all of this sort of whitish, pinkish color is blubber. And that is an essential part of the diet of Eskimos. And then there's far more muscle to go ahead with. But they'll use this blubber for energy. It is an important food product. They use it in place of oil or butter as well. You can make what's known as Eskimo ice cream out of whipped fat, or this blubber here, and wild berries in the summer. This is a close-up of what this series of baleen plates look like. And you can see they look almost accordion-like, that they're very closely packed. But there is a small space in between each one of these plates. And all of this fringe here, think of it like the edge of a broom. It can actually sort of collect and trap all those little microscopic organisms that these animals can then feed on. So I've talked about a good bit about the different types of food. I wanted to give you an idea really quick about what we can do with something like that baleen plate. And in this case, we're looking at a variety of contaminants, also known as heavy metals. These all naturally occur on the planet, but for a variety of reasons, whether they're anthropogenic or human-induced, or they naturally occur, say, volcanic eruption, you can get some concentrations that are very low, like you see here along this x-axis, the horizontal line at the bottom. And then you can see some here that are very high, much like this red line or these green lines. And they can represent different metals. And you can see it's not always the same concentration. Sometimes there's greater amounts. So what we did is actually took this baleen plate, and if you look from left to right, and notice it says summer 1987, winter 86, summer 86. Going back through time, we know the growth of that baleen plate. We can actually determine, wow, this peak here that we see in selenium actually occurred in the winter of 1984. Gee, I wonder what happened in the winter of 1984. So we can go back and look at the record of events that have taken place on the planet during that period of time. This is also another part of whales in particular that are called earplugs. They're basically the wax that builds up in the ear of great whales. But if you look here, you can see that there are all these different layers, sort of like we looked at in the tooth, the growth layer groups. Well, earwax in a whale is laid down like those tree rings, like those growth layer groups in teeth. And you can, and it's very fatty, it's lipid rich, and you can basically carefully unroll that. It's like sort of like rolled up like a cigar, as you can see. So you just sort of unroll it, and you can do these chemical analysis along the length and determine different time periods and what might have been taking place. We also are fortunate enough that hard parts, in this case, bone, this is the skull sitting upright, so that the very top of this image is actually sort of the tip of the mouth or the head of a whale. And this part here at the bottom of the image is where the backbone or spine would come in to the brain case of the skull. So bones have been collected through time, and we can get a suite of different information about what's going on and what's being recorded in the bone. We can get different metals. We can get different, what we call the stable isotopes here, telling us about their food, telling us where that food was collected. We can actually get some radiocarbon dates to actually get the age on these animals, because we don't always just work with modern animals. We can actually work with animals that have been dead for thousands or millions of years. So we can get paleo information as well. So this image is actually taken at the warehouse, one of the warehouses or storage buildings at the National History Museum, the National Museum of Natural History at the Smithsonian Institution in Washington, D.C. So this is a skull here on the far left, and this is a great image of the marine mammal curators and their staff, because it gives you an idea of just how large just the head, in this case, of a bowhead whale is compared to a human. And on those yellow shelving racks are the spine, the backbones, the vertebrae that make up the back of great whales. So we can use little bits, and it just takes tiny bits of sample. So what can all of this really tell us in practical cases? Let me tell you a quick story. In the North Pacific Ocean in the 1960s, so that would be the upper image. This is photographs from commercial fishery ships. The North Pacific Ocean was dominated by shrimp. That was the major fishery in the 1960s and into the 1970s. But in the 1970s, they started to see in their nets these larger fish, and they weren't lipid rich. They were very white meat, so not fatty fish, not lipid rich. And then by the 1980s, there weren't any shrimp. There were no shrimp to be caught commercially, and it was strictly these very large fish. So something happened, but it happened and it affected the entire ecosystem in the North Pacific Ocean. There are naturally occurring events that take place on our planet. This is not to say that there aren't human influenced events, much like we hear the term climate change, global warming. Those terms are often meant to infer human affected changes on our planet. But there are also naturally occurring changes that take place, and some of them take place on a semi-regular scale. So one of those is known as the PDO, the Pacific Interdecadal Oscillation. And basically what it does is just tells us, it records changes in sea surface temperature. Is it getting warmer? Is it getting cooler? The takeaway I want to show you is here on the bottom part of this image is sometimes you see these dark bars above the line, and sometimes you see these gray bars below the line. What they're saying is sometimes there are periods that it's warmer, sometimes there are periods that it's colder, and it can go back and forth. This image here, I don't want you to get all caught up and worried about the data. Mostly what I want to get across is that each one of these lines or data points are different colors, whether they're the brown or these blue dots or these pink dots or these purple dots, and some are connected with lines, and then there's the turquoise. They all represent different information from either the marine environment in the North Pacific or the immediate coastal environment of the North Pacific. And they can tell us different things. One is telling us what the sea lion ate through time. Another is telling us what microscopic organisms are found, and when they've died, they've settled down to the sediment in the ocean floor. Others are tree ring data, but they all can tell us information about what's going on in the ocean. So I just want to finish up by pointing out a few things. I just told you that there are naturally occurring cycles. Humans have been changing much on our planet, certainly in the last 200 years. What this image here represents is nearly 7,000 years. And if you just look at the white lines, these sort of going, here they go down in the fuchsia, then they go up, then they go down. Same in the purple or in the blue dots. They go up, they go down, they go up, they go down. Humans weren't affecting their environment on a global scale 5 and 6 and 7,000 years ago. This stellar sea lion and this series of stellar sea lions are telling us what was going on in the marine environment 5, 6, and 7,000 years ago. And we're getting that through the bones, much like those bones I was showing you stored at the Smithsonian Institution. We can also see these oscillating kinds of trends that can be collected and stored in the sediment. Everything's going to settle down to the bottom of the ocean floor. And if we take something like a sediment core and collect this information, we can go back through time and get a history of what's going on our planet. So why is this important? Why do we need to know the history? Well, this image is of a Native Alaskan family, and they have 2 seals on their quad track. And they're going to use every part of those animals. They're going to eat the animals much like you might have a steak or hamburger. Okay? And they're going to use the skin, and they're going to make boots. And they're going to use the bone, and they may do crafts, and they may weave baskets. And so that is probably the epitome of sustainability. You use every part of what you can use. But now these people also have to be concerned are they also eating contaminants when they eat that seal? Just like we have to be concerned, did I rinse off any pesticides that might have been lingering on my lettuce or my radishes? So with that, I'm going to stop. I hope this was able to give you a little bit of an idea of how we can use plants and animals, not just to appreciate them for the role that they play on our planet and in our ecosystems, and as well for their beauty, but also for the information that they can provide us and share with us. So with that, I'll see if anybody has any questions. And I thank you for inviting me this evening and for your time.
Thank you.
Awesome. Thank you so much. It's going to come out wrong, but I enjoyed that more than I thought. I don't think going into it, I quite knew what you were going to cover, and I found that very interesting. So thank you.
You keep your audience guessing when you come up with a title.
Yep, I like it. Before we ask you a couple of questions, we'll just, if my slides will progress, remind folks where they can find the recordings. So if you enjoyed this recording and you want to find more, you can go to our website and just look under that education tab at dolphincommunicationproject.org. You'll find the webinar section, and you can also view all of our recordings on YouTube. Our channel is cleverly titled Dolphin Communication Project. And once you have gotten all of the deep dives like this, which are geared toward an older audience, you can also tune into our dolphin lessons that are have the younger minds in mind. Then you can also check out our podcast, The Dolphin Pod, wherever you get your podcasts. And then, of course, we are happy to offer these programs for free, get as much of this accurate scientific information out there as we can. We would not be able to do that without our guest speakers. So a huge thank you to you, Amy, for doing this tonight. And then, of course, all of our supporters. If you enjoyed this and want to help us continue, you can support DCP by adopting a dolphin. You can become a member. You can even join us in the field. You can select DCP as your charity at Amazon Smile. And even if you don't select DCP, if you shop on Amazon, every purchase should start at Amazon Smile. Get there get them to spread their money out a little bit. And of course, stay in touch. So there you can see our website and all of our social media. So with that, Kathleen, did you have any questions that you wanted to start with?
I did. Well, one of them was just a question of interest. When you showed the ice flow, was there a seal off to the left in that photograph?
That was a Waddell seal.
Okay, I thought, I was like,
That was the real purpose that we were there for.
cool. And it's not, I mean, you don't hop from ice flow to get out to the.
No. And that is one reason I actually like that image, is that it's sort of a rare thing that you're not always, it's not like the ship's captains and the chief scientists like to let you out necessarily on those ice flows. It was not quite as broken up at that point. It was much more open water to that point, and so you can take inflatables like Zodiacs.
Nice. Well, the question, that was a question, but I was interested when you showed the piece of permafrost that broke off, and you were talking about how you can sample and look at these and get ideas of their content and maybe their chemical signature or pollutant signature. Is it possible to get that signature and then to trace it as it moves through the ocean? I mean, I'm not sure, but I didn't know if you could see where those signatures go.
It's hard. I mean, that's a really good question, because we do try to think about how finely can we time something, or do we know that this piece of land, this permafrost that has fallen off into the ocean, represented the year 1865.
Yeah.
We oftentimes can't do that, but let me relate a little bit to you how we've recently actually used this for more practical circumstance. We actually took some of the baleen plates from a harvested whale that was collected in a series of them from the 1960s through to the year 2000. And we did some of these chemical analyses along the length of the plate. And we were able to see that there were changes in this increasing in the amount of these heavy metals, particularly once we started getting into the early and the mid and even the late 1990s. We're like, gosh, what could be doing this? They're a migrator, so they go back and forth from one location to the other location every year for their entire life.
Wow.
And we're like, what's going to change here? What's changing? And then it finally occurred to us as I started looking at the data and going, the permafrost really started melting. We really started noting it in the 1990s. And it's only just been increasing more and more. And that seems to coincide with this increase in these heavy metals.
Wow.
So we can't say in that case is like, this is 1986. We do have some idea working. And again, this is where you have to collaborate with experts in other fields. In this case, we work with hydrologists and glaciologists to get an idea of if your glacier has melted back 25 meters, what kind of time period is that?
Mm-hmm.
And so we can get an idea of these sort of larger groups of time.
That segues nicely to a question that I got, which is about the collaboration. So a little bit of maybe the easy parts, the fun, if you will, parts of all that collaboration, and then some of the challenges. And specifically the figure you put up that showed all of the tracking data from so many different species. Can you give listeners a sense of like how many groups went into creating that?
That was a lot to put it very broadly. We had, I believe there were well over 24 different organisms on that plot itself, a lot of that tracking data was gathered with persons at federal agencies. Most of that came from collaborators within North America, whether it was Canada, the United States, or Mexico. And then also picked up and working with our colleagues in Russia and Japan, as well as South Korea. I would probably, if I had to put a number to that, I would say there were easily 100 people involved over time, collecting that kind of data. And as you know, I mentioned it earlier in your question is sort of that twofold. It's like, yeah, this is great. You want to collaborate. You want to work with people who have diverse backgrounds. You can't be an expert in more than one. You're lucky if you're an expert in one area, let's be honest, right? And you have to work with other people, and because you can't be all places at all times, and you can't do all things at all times, and nobody has those kind of resources. So that's one of probably I guess some people might find it really challenging. It's like, oh God, I got to work with all these people. But in a sense, I love it myself, because I can talk to my colleagues in Russia. I met new people in South Korea in this past year. I was asked to be a pinniped expert for the North Pacific Ocean. And this group was out of South Korea. It was like, how did you even know about me? Which is that's a really cool thing. I know people literally on every continent on the planet. And yes, while nobody actually lives full-time in Antarctica, for the most part, there are all kinds of people working and living in Antarctica. So that's the great side. That's the wonderful side of this. The challenging side is I've literally been up at 1 a.m. in the morning to make a meeting with colleagues in Singapore or Australia, or trying to figure out, how do I get this piece of equipment or this crate or this case of instruments into the field. And I gotta say, probably one of the more complex and challenging things is to make sure you have all your permits, because what might be permitted in the United States is not necessarily permitted in the Bahamas. It's not necessarily permitted in Portugal. It is not necessarily permitted in South Africa. And if you're going off to work, like a colleague of mine is right now in the Seychelles Islands in the Indian Ocean, he's got to think about all the points in which his equipment has to pass through just to get there or get back. Really quickly, I was party to a specific walrus cruise that was leaving various ports in Alaska, crossing into Russian waters, back and forth across the Bering Sea, started in the US, ultimately ending in Russia. None of our samples were allowed to come back to the United States. You sometimes just have no idea what the geopolitical situation is going to be. And that was a lot of resources in 2 months' time just on the cruise.
Wow.
Yeah, that just causes you to cry.
Heartbreak.
Yeah.
Yeah.
Wow.
And those are things you'll just never be able to replace.
Yeah.
Yeah.
But it is the nature, you don't have to be a scientist to have things like that happen to you.
True.
And you just go, okay. And it wasn't the scientists, it was people higher up in the political realm that wanted to make a statement, and that was their way of making a statement.
But the animals don't pay attention to politics or boundaries. So it definitely is
Not human.
a challenge to study them.
Yeah, that was probably the most interesting thing. And I'm so glad I had gone to Alaska to do that research, and I ultimately ended up being there for 14 years, is because you're so close to Asia and including countries like Russia and Canada, and learning to work in environments where maybe you're fine or the US is fine working with their Russian colleagues, but the Russian and the Japanese aren't getting along. And it's not the scientists again. The scientists are all generally fine because you're choosing to work with each other. It's the, sometimes it's the agencies, sometimes it's the, you know, higher ups, the governments, it could be. And that's a challenge, but that's okay. Life is full of challenges, and it's about overcoming the challenges that really makes things worthwhile. So you start to figure out, you just learn to adapt, and it's like, how can I get this done?
Nice.
Awesome. Well, thank you so much for leading this talk. And we know it's a great addition to our collection. And we hope everyone who's been listening has enjoyed it and will join us at another program. So
Thank you.
good night and thank you.
Thanks for inviting me. I really appreciate it.
Thank you very much. Have a good night or good afternoon when you listen.
Bye.
Bye.